Here we come with part 2 of histopathological examination. In this video, we are going to discuss the objectives or steps which are included in histopathological examination. So let's get started. Following are the steps included in histopathological examination. First step is sample collection, second sample transport, third sample receiving and labeling, fourth grossing, fifth tissue processing, and the sixth step is microscopic examination. Now the first step, sample collection. This is the initial step in histopathological examination, which is done by OT staff in operation theatre. The knowledge of histopathology should begin from the operation theatre, where the samples are collected after an adequate patient identification. Patient identification, sample orientation, and an adequate fixation is the responsibility of the operation theatre staff or OT staff. Tissues are collected and fixed by an appropriate fixative, and for this 10% formalin is the routinely used fixative in histopathology lab. 10% formalin. But few tissues require special fixative, as for example Boyne's solution, which is used for testicular biopsies because testicular biopsy is fixed better in Boyne's solution. Now another important point is tagging. Tagging of the specimen is highly recommended, which is also done during or after collection. Now sample transport. This is the second step in histopathological examination. This is the process of taking the samples from the operation theatre and transport it to the respected histopathology laboratory. Sample must be fixed in adequate fixative or 10% formalin, which is a routinely used fixative in pathology laboratory, as we all know. Samples may be transported at room temperature in properly labeled and well sealed wide-mouthed bottles. Sample receiving and labeling, which is done by the laboratory. This is a very important step performed by the laboratory and should be done very carefully to avoid misidentification errors. The specimen is usually sent in a wide-mouthed bottle which is filled with 10% formalin. The specimen or sample which is received is already labeled with patient details, so it is the duty of the laboratory to cross-match the information from the container to that TRF form and note down their data. And the data should be registered in the laboratory register. Fourth step is grossing. Grossing is the bare or naked eye examination of pathology specimen with cutting and tissue sampling in order to prepare material for subsequent microscopic examination. So, grossing is a process in which the specimen is inspected with bare or naked eye to check any visible abnormality, and then cutting is done accordingly for subsequent microscopic examination or to prepare the material for subsequent microscopic examination. It is an initial step before tissue processing in histopathology for obtaining accurate diagnostic information while being further processed for microscopic examination. After grossing, tissue undergoes for tissue processing. Now, what is tissue processing? Tissue processing is a technique by which tissues are made suitable for sectioning or cutting, followed by embedding. Tissue processing is done by two methods. First one is automatic and the second one is manual. For automatic tissue processing, there is a machine automated tissue processor which is used, also known as ATP, and the manual tissue processing includes following steps. So the steps includes in tissue processing are: first is fixation, second is dehydration, third clearing, fourth infiltration and impregnation, fifth embedding or block making, sixth microtomy or section cutting, seven staining, and the eighth one is mounting. So the first and important one is fixation. Fixation is a chemical process by which biological tissues are preserved from decay. Why it is so important? Why it is so necessary? Why we done fixation? And the answer is, when the tissue is removed from the body, the blood supply to it is cut off. As a result, the cell undergoes complete disintegration due to autolysis and putrefaction. To preserve the natural state of the tissue is immediately placed into a solution called fixative, and this whole process is called fixation. The common cause of poor cellular details in histological preparation are due to autolysis and putrefaction. Autolysis or self-destruction is the death of cells due to action of enzymes which are liberated as soon as the cells dies. Autolysis is the lysis of the cells by the enzymes liberated as a result of rupture of enzymes, which is lysosomes. Whereas putrefaction means the breakdown of tissues by bacterial action, often with the formation of gas. Both the autolysis and putrefaction follows rapidly upon death of the cell and after removal of the specimen from the body. This causes the disappearance of nuclei, cloudiness of cytoplasm, and the cell loses its staining properties. These changes of the cells can be prevented by adding some chemical substances called fixatives. So fixation is a process by which gross and microscopic anatomy of the tissue is preserved. And the second one is dehydration. Dehydration is the removal of water from the tissue. Now the question is, why we remove water from the tissue? It is important because water is not miscible with paraffin wax, which we are going to use in next steps. Dehydration is done in few steps, from lower concentration alcohol to higher concentration alcohol. As you can see, 30% to 50%, 50% to 70%, 70% to 90%, and 90% to absolute alcohol. We have to go to step by step or in increasing concentration, otherwise sudden change in alcohol concentration can cause damage to the tissues. So the dehydrating agents are alcohol dioxane, acetone, triethyl phosphate, but majorly dioxane and alcohol majorly used in histopathology lab. Now the another one, next step is clearing. This step is for removal of dehydrating agent to make tissue suitable for the diffusion of paraffin wax, because dehydrating agents are not mixable in paraffin wax, but there is an exception, which is dioxane. Dioxane is a dehydrating agent which is miscible with both water and wax, but others are not, so we have to remove the dehydrating agent by using the clearing agent, which are miscible in paraffin wax. Clearing agents are xylene, toluene, chloroform, benzene, cedar wood oil. Clearing makes the tissue transparent, which increase the refractive index for microscopy. Now the other step is infiltration and impregnation. Next step is infiltration and impregnation. Infiltration means removal of clearing agent, whereas impregnation means diffusion of paraffin wax. So the removal of clearing agent is called infiltration and the diffusion of paraffin wax is called impregnation. It provides internal strength to the tissue. It is done by keeping the tissue section of autopsy or biopsy in a jar of molten paraffin wax. The melting temperature for paraffin wax is 56 degrees Celsius. It can vary to 56 to 58 degrees Celsius depending on the quality of paraffin wax. Now the next step is embedding. Embedding, also called as block making, it is a process to give the tissue external strength or hardness to make it suitable for section cutting by making a square block. In previous time, we use L mold of brass metal, which are two L-shaped mold, but now plastic assets are used for block making. Now the embedding medias are carbowax, paraffin wax, paraplast and others. But paraffin wax is mainly used because it is less expensive. Now blocks are ready for microtomy. Now the next step is section cutting or microtomy. It is a process to cut thin ribbon-like slices of tissue sections by a machine called microtome. Before section cutting, there is one more step which should be done if necessary, that is trimming. Trimming is the process of removal of excess paraffin wax that is sitting around the tissue, which expose the tissue in order to be sectioned. Post trimming, the tissue is cut into thin slices and placed on glass slide and then fixed onto a glass slide. After the embedding of tissue section with paraffin wax, these sections are processed for trimming process. Insert the paraffin block into block holder of a microtome. Move the block forward so that the wax block almost touches the knife. To trim away any surplus wax and to expose a suitable area for tissue sectioning, the section thickness adjusts to 15 microns, and then trimming is done by microtome. And then after trimming, we done the microtomy by using the same microtome. After trimming, there is a point flotation bath. So dissected needle is used for splitting tissue section and picking onto the slide so that tissue can be fixed. I mean to say, the tissue section is picked from the slide by dissecting needle, and then tissues are placed on the flotation bath which have water in it. This process is done to remove the wrinkles or foldings from the tissue. After this, there is an important step which is deparaffinization. The deparaffinization. After this, the section which have paraffin on their surrounding goes for deparaffinization by slightly heating the slides on hot plate. After deparaffinization, slides are dipped in xylene jar because paraffin wax is miscible in xylene, and we have to completely remove the paraffin wax. So we can remove this wax by xylene. Now the next important step is staining. Before staining, we have to done an important step which is hydrating the tissue. It is not dehydration, it is hydration. Dehydration, which we done in second step, you can see paraffin is not miscible or soluble in water. That's why we remove water from tissue in the second dehydration step. But now, because our stain is aqueous in nature, so it required hydrated tissue for staining. So we need water in tissue section, and this step is called hydration. Now we have to remember one thing, that this step is hydration, not dehydration. In dehydration step, as you can see, we used increasing grade of or concentration of alcohol, but in hydration we use its opposite. Now we use higher to lower concentration of alcohol, and lastly distilled water. So the steps are 90% alcohol, then 70% alcohol, then 50% alcohol, then 30% alcohol, then distilled water. And then after hydration step, we done our staining. So staining is used to highlight important features of the tissues. Mainly H&E staining is routinely used in histopathology lab, which means hematoxylin and eosin stain, because hematoxylin is a basic dye, so it imparts blue color to the nuclei, and eosin is an acidic dye which stain the cytoplasm into pink color. There are some special stains also in histopathology, as PAP stain, PAS stain, reticulin stain, and others. Now for further process, we have to remove the water again. So the next step is dehydration, which is same as in the second step. We have to use alcohol in increasing concentration from 30 to 90%, and then absolute alcohol. Then after dehydration, we have to done the same step. That was the third step, clearing, which is done with the help of xylene to make the sections suitable for mounting. So the eighth step is mounting. Sections are mounted in mounting medias. Mostly used mounting media is DPX or Canada balsam. DPX is stand for dibutyl phthalate xylene. This step is important, or this step is done for future studies because mounting media preserve the cellular detail of the tissue section for long time. In mounting, we make a square shape around the tissue section on the glass slide by the help of mounting media, and then cover it with cover slip. So This is all about mounting. Then the last step is microscopy. Now tissue sections are ready for microscopy to diagnose any abnormalities or for clinical diagnosis of cancers. So these were the steps which should be followed for routine histopathology examination. If you want detailed information about each step, then comment in the comment section. Stay tuned.